Good evening, ladies and gentlemen. My name is Savio Wu, and I'm kind of moderating the program for tonight. And we have lined up three wonderful speakers for you and trying to relate to you what has been the most exciting progress that the scientific community has made in the area of gene and cell therapy for cancer. So I think all of us are in for a real treat tonight. And thank you for coming. The three speakers tonight is Professor Carl June from UPenn, Professor Larry Cooper from MD Anderson, and Michelle Satterlane from Memorial Sloan Kettering. And they each have a wonderful story to tell you, and I'm not going to, I don't need to tell their story for you. They will themselves, and very nicely. I just want to say that, don't forget about reading the words on the slides. Even I cannot see them. The important thing is just to look at the blue. 23 million is the total dollar that ACGT has raised to support research. And ACGT is a 403B foundation founded by Edward and Barbara Netter about a dozen years ago. And the only mission is to raise funds to support research that can be translated into the clinic so that the patients can be helped. And over 12 years or so, we've raised $24 million, and it's still going very strong. The important thing is that in many charitable foundations, only a fraction of the dollar that is donated actually goes to support the mission. Here in ACGT, 100% of donations goes to direct support of research, 100%. All the administrative costs is covered by another source of income, that an anonymous donor. And we all know who that is. 17 is the research projects of clinical translational research projects that has been supported by ACGT. That's a significant number of clinical trials that's ongoing, only because ACGT was able to support them. Otherwise, this program would not have gone forward. So this is a major impact that the foundation is making in terms of trying to get the field forward so that we have more cancer patients that can benefit from this groundbreaking research. 3 is our 3 award-winning doctors here, and 1 is ACGT is the only charitable foundation whose mission is to do only gene and cell therapy for cancer and nothing else. So these are the critical numbers. Anyway, so I've taken up enough times, and I would very much like to introduce you to the 3 wonderful speeches that we're going to hear tonight. The first one up is going to be Professor Carl June. He's from the University of Pennsylvania, and when his grant first come forward to the Scientific Council, we all thought that this is kind of out of the left field, and we had a long discussion about whether we should fund this kind of research or not, because it's kind of iffy. And so finally we said, well, the scientific data is so good, it's so promising, let's give it a try. It's only Edward and Barbara's money that we're spending. And so we did, and here we are, Carl.
Thanks, Savio. In front of us are, this is a cottage industry of what we'll call CARs, chimeric antigen receptors, and it would not have happened were it not for the Alliance for Cancer Gene Therapy. So Michelle Satterlane and Lawrence Cooper have been leaders in this field. There's a group of 5, you can count on one hand, in the country that have done this. It did not happen in the pharmaceutical industry. It was completely done in the academic industry, but it was also not done by the National Cancer Institute. And that's the bottom line message. It was done through philanthropy, in my case, and Michelle Satterlane's, as you'll hear. We got grants in 2004 that allowed us to do our trials, and I also had funding from the Leukemia and Lymphoma Society, but not the National Cancer Institute. It was too far out there. And where we've been with cancer is a history of starting with therapies that were really non-toxic and have a lot side effects, so-called chemical chemotherapies. And then in the 1990s, the first biologic was licensed by Genentech. That was rituximab for lymphomas, and then now we've had blockbusters like Herceptin for breast cancer. These are much better than the chemotherapies, but they don't cure people. They cost about $100,000 a year, and but they do have much less side effects. What the Alliance for Cancer Gene Therapy is doing is to fund, gene and cell therapies that have the potential to be both in the long run cheaper and curative, to not have the iterative and recursive administration that we do, with the biologicals that the biotechnology industry has done. So I think we're at the dawn of where there's a new industry, which will be cell and gene therapies. And I'll tell some of that story and you'll hear more. Mine started back in high school when I was a football player. And this is a picture when I was trying to chase Kiko Garcia. I was number 75. Kiko played 10 years on the Baltimore Orioles, and he beat me. And then, so that was my high school career. Then I was going to Stanford in April, I was admitted in April 16th, 1971, but then I got drafted and went from the West Coast to the East Coast to the Naval Academy, and I thought I would play football there. What I discovered was that some people kept getting stronger and some didn't. And I hit a plateau. And what I found, and when I actually started medical school, the medical textbook said that steroids don't work. That they don't make you stronger. What they said is they made you swell up, that your muscles gained water. And but I saw firsthand people who took steroids and got stronger. So I said, sometimes the medical profession's not right. And then I graphed Barry Bonds' home run hitting records. This is the first real example in the professional where you could really quantify what is now called synthetic biology, which is making the neuromuscular system better than it was. And it showed how many times he had to go at bat here to hit a home run when he was a rookie, and he took 25 to 30 at bats. Then he, got into the swing of things. He was a mature athlete, one of the best home run machines ever, over a period of 10 or 12 years. And then something happened that never happens when you turn 40. He got better. And he went down to only taking 4 or 5 times at bat to hit a home run. And that's when he started drugs. And so what my lab has done, and the people you'll see here, has been to work in this area of synthetic biology. In this instance, can we make the immune system work better than nature did it? So our immune system has evolved to make decisions to get rid of viruses, but it's not very good at controlling cancer. It usually fails. So that's the idea, is can we put the immune system on steroids? And you're going to hear some very clever ideas on how to do that. So synthetic biology is this idea of, instead of using evolutionary biology, which is how we got an immune system that's good for the flu, how can we design it in an engineering way to make it better than nature did? And there are different aspects of this now that are just beginning to enter into daily life, such as artificial bacteria have been created that can eat oil. And I think you'll hear the story from all three of us of so-called CAR T cells, which are the first example of synthetic biology in clinical medicine. So T cells are a form of white blood cells that we have. They're in your blood and primarily in lymph nodes and spleen. They can last your entire life. So that's why when you're a child and you get a vaccine, your immune system can last the rest of your life, basically, against that. They make smart decisions. They can sense their environment that they're in. And most importantly, a T cell can kill a tumor cell that's resistant to chemotherapy. That's called non-cross resistance. They kill in a very different way than chemotherapy does. The problem is that there are no T cell based adoptive therapies that are FDA approved. So this is a transfusion therapy where you have to take a patient's cells out, grow them, modify them in the synthetic ways we'll talk about, and then give them back to patients. That's been, people have had these ideas for more than 20 years, but they haven't ever made it to FDA approval. I think we're getting close. So the general approach of this kind of CAR therapy is shown here. In a way, it's a blood transfusion, and it's like a bone marrow transplant. So basically, a patient donates their cells from a blood transfusion to the blood bank. They're then manufactured in several ways, and you'll hear we each have different twists on that, but the same thing happens in that this CAR construct goes into the T cell, and it then stays there permanently. Then the cells are grown for a period of time in the lab, about 10 days, and then given back to the patient as a simple blood transfusion. So that's the actual process. And the issues on this is really different than what the pharmaceutical industry does, where they make one drug that treats all. These are customized, patient-specific vaccines, which meant there was a market failure here. Because of the disappointing history, the biotechnology industry wouldn't invest in it. They thought it was a waste of time, and it wasn't worth this complex logistics. And the first thing was we needed to build a T-cell culture system, because unlike blood cells that are transfused every day, those, we have universal donors. For your T cells, unless you have an identical twin, you have to be the donor. So that means you have to figure a way to do this, to grow your cells out in a laboratory. And Bruce Levine in my lab began working on that over 20 years ago, and we made little beads that grow cells. And these are the beads that have iron in them, so they're paramagnetic, and we can remove them. While they're touching the T cell, this is a T cell. They grow, the cells grow, and they'll grow up to 1 million-fold in a period of a few weeks from one starting cell. And then in the end, we remove them, using a magnetic field. And then these are pure T cells here. That's the transfusion that's given to the patient. So we started this work initially testing in HIV. And that's because ethically, patients, back then who didn't have T cells were HIV patients because the virus kills them off. So we found that it was safe to transfuse the cells in patients. Now, a CAR cell stands for chimeric antigen receptor. And so we have here a T cell that has the T cell receptor. Then T cells evolve to kill viruses. So this may be, say, a flu-specific T cell receptor here. This is a bad guy. That's a tumor cell, and it has a target here called, in this case, CD19, which is on some leukemias. But this does not have a receptor on this T cell for the tumor cell. What we then use is a vector. It's an HIV-based virus, which then introduces a gene into that cell permanently, because that's HIV evolved to introduce genes into cells. But this is a gutted form of HIV that doesn't cause disease. It can only infect a cell once. And then what happens is this T cell now starts to make from the new gene that it has, the chimeric antigen receptor, which is on the surface of the cell. And this now can bind this target called CD19 on the leukemia cell. So now this T cell has become dual specific. It can become still a flu killer here. It still has its original T cell receptor, so it may be for protecting against flu or smallpox or shingles, and it also has now a receptor that can then, when it binds to a leukemic cell, it will bind to it and then kill it. So that's the principle of how these work. This T cell then, after it's killed its target cell, can then go on and it will divide. And that's why they're called serial killer cells. So one T cell can divide many times in a patient, and that's necessary if you have a lot of tumor cells. So the actual process of these CAR trials is shown in this cartoon. There's 4 or 5 steps depending on the protocol. So initially, as I explained, the patient has blood drawn, and then there's this 10-day manufacturing time where the CAR is introduced. Then the cells are, the beads are removed, the cells are frozen, and then the cells are given to the patient. In some of the protocols, the patient gets chemotherapy a few days before the T cells are infused. In other protocols, they don't. And the initial trials that we've all tried here that you'll hear for ethical reasons were done in patients with refractory leukemias and lymphomas. And that was because the target CD19 and CD20 that's on these B cell targets, leukemias, is not on any other tissues that you can afford to not have to lose those cells. So if you target, for instance, a lung cancer, and that target is also on your normal lung cells, you're going to be in trouble because you won't have lungs. The T cells can't tell the difference between a normal cell that has a target and a tumor cell. All they care about is, do they have that target? And so we started where we knew it was safe and we could target CD19 and on both acute leukemia and chronic leukemia. This is a disease that occurs in children and adults. We've treated children, that Michelle Sadelain has been treating adults and now I think children. And this CLL, we both have been treating. It only occurs in adults. It's very rare that anyone younger than age 40 gets that disease. And these diseases are not curable without bone marrow transplantation in general. And in CLL, most people are too old to have a bone marrow transplant. So they're destined really to take a lot of different chemotherapies as a current therapy. So we reported from the Alliance Cancer Gene Therapy trial that we were funded in 2004. We treated our first patient in July of 2010, and then in August 2011, the first 3 patients were reported. And all 3 patients had incurable late-stage advanced leukemia with huge amounts of tumor, 3 to 7 pounds of tumor in each patient. So this is a tumor that grows slowly, but it grows over many years to big masses of tumor. And what we found was that each patient responded, and that each of these infused CAR cells could kill about 1,000 tumor cells because we could calculate how many tumor cells the patients had. And we now know these remissions are durable, so the first 2 patients that had complete remissions remain in complete remission. They haven't had any further therapy now, 3 years later. And we also found these CAR cells have been sustained in the patients, and they're continuing to make these antibodies that bind to CD19 in the patients. So then after we reported those initial 3 patients, our 4th patient that we treated was a pediatric patient, and we call that our PDCAR trial. But anyway, you're not allowed ethically to treat pediatric patients until you've shown that a therapy can work or has safety in adults first. So we had initially tested our CAR trial cells in pediatric leukemia in mice. That was what we did. And then we began in August, or actually April of 2012, to treat children with CARs. And there are different kinds of CAR designs. We first began treating patients actually with HIV with a so-called first-generation CAR. And this is a schematic that Michelle has made. There's a lot of vector, very fine design issues on how to make more potent CARs. We're right now basically testing what the field's calling second-generation CARs or sports CARs, but we can make them more potent than what they are now. So there's a lot of different next-generation ideas that are out there on how to enhance this. So our first patient was reported at the hematology meetings last year. She was treated in April, in April 16th, and this is what she looked like then. And then the hematology meeting, she was reported at that time. She was about 6 months after treatment and in remission and looking much better. And she was on the front page of the New York Times then. She was then finally reported in the scientific literature in March of this year, at the same time that Michelle found really striking results in adults with ALL. So this is a disease that kills you in weeks or months. It's not like chronic lymphocytic leukemia that can oftentimes take a decade, of natural survival with the kinds of therapies we have. And so we didn't expect it to work as well because this tumor grows much more rapidly than chronic lymphocytic leukemia. And what we found is you can learn a lot from one patient. This patient really got much sicker before she got better. She had 106-degree fevers. And what that was we found out it wasn't an infection. It was her immune system killing pounds of tumor. But then she got better. When the tumor went away, the fever went away. So we learned that, and that's been a recurring theme now in our patients. And we also found that cute kids attract a lot more media attention than adults, particularly old men. So anyway, this is where we are right now. This is Bruce Levine, who has worked with me for about 25 years on this. We've treated 55 patients, 20 kids and 35 adults with chronic leukemia. Had very high response rates. That's really pretty much unheard of for phase 1 trials in patients with advanced incurable cancers. And but the issue then is, how do we treat more than 55 patients? We can treat 4 to 8 patients a month at the University of Pennsylvania, and I think it's probably similar here at Memorial and at MD Anderson. How can we scale up to treat, because there's 15,000 cases a year in the US alone. And so the answer is, I think, is that we need to have a new healthcare industry. Right now what we have is the pharmaceutical industry, who makes drugs that one shoe size fits all. We have the biotech and then medical devices industry. And what will happen if this continues to be successful is that there will be a new industry, as I was mentioning, of cell and gene therapies. And that requires, as I showed you, a very different business model. It's sort of like the FedEx thing of where it may, at least at this point, it'll start as a patient, go to a manufacturing center, and then come back. So that's how this is different than standard drugs. It's each particular patient, it's called N of 1. Each one is an FDA-approved product, each patient you treat. And can it be done in a blood bank or in central manufacturing? And all those things are being worked out now. And there's an analogy then of how do you scale something up like this. Right now, our cell culture process takes PhDs to do. It takes people to make intelligent decisions on what to do, when to feed the cells, et cetera. When Henry Ford first started building the Model T, they had manufacturing lines, and they had technicians that would assemble cars, and they specialized. And now cars in Detroit are built with robotics. So if you have a simple car, then about 80% of the car is assembled with robotics. So what has to happen, really, I think, is that these cell culturing systems that we have to become automated. And that's not something we can do in the academic arena. This needs to be done in the pharmaceutical industry. And there's been no effort on this at this point because we've been working on shoestring budgets at the academic centers to make the actual treatments, but there's been no effort at scaling this up to industrial scale, which is what the next step is. So there are some lessons on how to do this from engineers. This is some examples of a review article of, if you have a very fixed cell culture system, then it's much easier to automate it than if you have to have a system that's highly customized. So if you have a Volkswagen that has no options, that can be made essentially 100% by robots. But if you have a very customized car, such as a Lamborghini, that's always going to be very expensive because it has to be put together by hand. So last year we had the entry of the pharmaceutical industry into now making these robotic large-scale systems. And in August, Novartis announced an alliance with the University of Pennsylvania to this. Celgene has just done this with Baylor down in Texas, and there's a number of biotechnology companies now starting up where there had been none, just 2 years ago. So we're finally seeing a way where we can have technology handoff, for these advances that are happening in the academic medical centers. So I've learned a lot about this. It's been about over 20 years that we've been working on this. You have to have a long-term view, in this case, be very stubborn because we got turned down a lot of times. We're finding that now these multidisciplinary teams that are best funded really by philanthropy and not by the NIH. The NIH funds stovepipes. It's a hierarchical system. It funds a little grant that funds one lab. What we need to do is have networks of people that combine their expertise, and that is in fact what philanthropy has been setting up over the last several years as the NIH has really become unable to really fund much. We've also found that there's a large need now to educate the public for new complex cell and gene therapies. And this cartoon came out at the time of these initial CAR trials were published. And my daughter who's in college actually sent it to me. But what it says is, what's the deal with these leukemia trials? You got to see helping the immune system attack tumors has been a long-term goal, but always lots of promising leads don't pan out. So what they do, they took some patients' T cells and patched their genes, using viruses like leukemia viruses or HIV. And then they got the patch added code to get the T cells to replicate wildly and persist in the body, which is true because the cells had to persist, the CAR T cells, to kill all the tumor cells. But then they said, it created its own set of problems. In our case, we used an HIV-based virus. So how did they actually patch the genes? They said, what specialized in invading and modifying T cells? Seriously, yep, must have been a fun conversation. So that's HIV. HIV evolved to modify and engineer our own immune system. It's just it could be made non-pathogenic by gutting it out so that you take out the disease-causing genes. And there are ways to do that, the viruses, so that they can be used as tools rather than pathogens. So yes, it's okay because we've treated the blood with HIV. Are you sure you're a doctor? Almost definitely. So that's kind of where we're at this point. We need to educate patients now about these new therapies. It's that we'll use your own immune system and complex gene manipulation things to make things that in the end are more targeted and less toxic and more effective. So I'd like to thank you for your attention.
Thank you, Carl. Because of the 3 lectures or 3 speeches tonight, I kind of employed the same theme of immune therapy for cancer. We're going to go through all the presentations and save the question and answer period at the end. Okay? And Larry from MD Anderson, he's going to tell you another aspect of cell and gene therapy.
So thanks very much, and obviously thanks to the Netters as well. I got my first grant from this organization back when I just started out in the lab, and it was sort of, without that grant, I'm not sure actually my whole career would have taken the trajectory it has. So a lot of trust at that time, and a lot of gratitude on my behalf. So I'm going to tell you the story that's somewhat similar to Carl's, but has like its own flavor. And I think it's important to realize that the United States does best when competing groups essentially go after big prizes. And a rising tide really raises all boats. Smart people, when they come to the table, energize other smart people. And that competition is good for the system. And I think just as the philanthropy money is important, so it is to have really fine scientists like at this table who are all working really hard to push this out, because competition is a good thing. So I'm going to tell you a little bit about better living through gene therapy, and I'll advance. So I'm just going to take a second just to kind of paint the big picture. You may not know some of these things, but it's worth just hammering home the cancer message, particularly in this time of sequester and budget impasse by our representatives in Washington, because this really is a national tragedy. So roughly speaking, about 1.6 million Americans will be diagnosed with cancer in the United States. In Texas, when I have these types of talks, I say that's roughly speaking the entire population of Dallas and Fort Worth coming down with cancer every year. Basically, every 20 seconds, an American is diagnosed with cancer. Every minute, an American dies with cancer. And then I put this in, obviously, because of 9/11 in New York. And that's basically 9/11 every 2 days for the entire year. Right? And we know we would basically bankrupt the country going after 9/11 and putting our national treasure on foreign shores when we have a national emergency here in the United States. So we have to consider our priorities, I would think. So the war on cancer has been going on a long time. It actually was started by Richard Nixon. So this was a good part of his legacy. And it's still, however, the major cause of death 40 years on for basically Americans. And it's actually not just an American problem now, it's a global problem. And you can see the statistics right here. 12.9 million new cases are diagnosed with cancer in 2009. And by 2030, which is just around the corner, 27 million of our fellow citizens on planet Earth will have cancer. And that will yield 17 million deaths, essentially, a year. From malignancies. So again, we have to address these types of problems, and the United States is in the pivot, is the lead country that can solve these types of problems. So as Carl alluded to, and this is my own kind of stab at it, the war on cancer has evolved sequentially over these years, and we started out with the bulwarks, the 3 major modalities of chemotherapy, radiation therapy, and surgery. And for a long time, that's what we have. But in the modern age, we've really come to recognize that we can have really targeted interventions. Carl mentioned rituximab, antibodies to CD20. There are actually other types of molecules, small molecules that penetrate cells and sniff out the genetic lesion in that cancer cell so that they can actually switch off that cancer cell or target that cancer cell to die. And those are known as these sort of targeted therapies, and terrific work has been done there. The third rung down is really almost where we are today, and the fourth is what's coming right around the corner, and that's this world of immunotherapy. And that's why you're here today, to hear a little about it, because you want to know what the future is going to bring you. And it's essentially this world of immune-based therapy and then combining gene and immune-based therapy to really make a potent product. So cancer immunotherapies, when we think about it as scientists, and as physicians, we see a whole catalog of different immunotherapies. And we're right now just in one small corner of a flowchart that just looks like a spider web. But when we think about immunotherapy, we think about how to manipulate the entire body's immune system. Whether we want to do it with vaccine, you all hopefully got the flu vaccine this year. If not, you should go down to Duane and Reed or whatever it is around the corner and go spend your $20. And that's an example of an active therapy where we would give essentially a vaccine to a patient. And then on the other side of the ledger is this world of T-cell therapy or adoptive therapy where we're putting cells into a patient. And all of this essentially is now in the marketplace and is available to us. And there's not a thing on here that we don't do at these major medical centers on the war on cancer. But for today, we're just going to focus in the red part and these idea of genetically modified T-cells and products. So T-cell therapy actually has a history that goes back to Don Thomas, who got the Nobel Prize for this, but he didn't call it T-cell therapy, he called it bone marrow transplantation. He got the Nobel Prize back in about the 1990s, and he just sadly passed away just a couple of years ago. And what Don Thomas taught us was that you could essentially move T-cells from one person to another. But at that time, he was moving whole bone marrow, all essentially the progenitor cells from one person to another. But the grafting of that bone marrow in a patient gave rise to these blooms of T cells, these blooms of immune cells that could sniff out friend from foe. And I'm going to diagram that for you. But that really is, as we trace it back, the hallmark, and still today, is the best example of T cell therapy we have today, is in the context of bone marrow transplantation. So bone marrow transplantation, in fact, we reword this now really to call it hematopoietic stem cell transplantation, is this concept of overwhelming amounts of chemotherapy that are given to patients. Lethal doses are given to patients, but they have to be resuscitated. And they're resuscitated by infusing in bone marrow or hematopoietic stem cells, progenitor cells, that restores the bone marrow function, and importantly, provides this new immune response, provides these waves of T cells as well as other types of immune cells that can target a patient's malignancy. And that little circle diagram, it goes to show really my career. So I didn't actually start out playing football. I started out playing rugby, but I was a very average rugby player. I was thoroughly thumped, to tell you the truth. But I live, and like all the investigators on this table, really live at the boundaries, because it's really at the boundaries of fields that we're really able to make the best approach. And I'm actually a pediatrician. I run a pediatric bone marrow transplant unit down in Texas at MD Anderson. So I live in one part of the Venn diagram between bone marrow transplantation and pediatrics, but then the other part of the diagram is in this world of immunology or immunotherapy. And it's trying to piece all of those three things together and live in the middle there of understanding immunology, understanding transplant biology, and understanding how to do this in children. So to tunnel in now, and you guys can go to medical school here for a few minutes, to look at how bone marrow transplant works. How would it work essentially for Don Thomas when he was working on this? So we think about bone marrow transplantation very much as sort of a yin and a yang. When T cells grow like seeds in a lawn in somebody's bone marrow because they've been, essentially that bone marrow has been gifted to a person, those T cells come out, those waves of T cells come out, and they essentially can be divided into two major categories. One side of the ledger is what we don't want. And this is a man-made disease called graft versus host disease. This is where the T cells trespass and recognize normal structures, and that person's injured. They can have whopping autoimmune disease. Skin can be reddened, their liver can be injured, their gut can be damaged, they can have overwhelming amounts of diarrhea. This is where we don't want to go. Where we want to go is on the other side, where we want those T cells to recognize just the tumor cell and leave your normal body, leave the recipient's body untouched. So how does this T cell make that decision? How does it make that, essentially that distinguished friend from foe? Well, it does it because of a very special molecule called the T cell receptor, and that you can see is red and blue on the top there. And it's that red and blue T cell receptor that actually interrogates and asks the question at the molecular level whether or not the cell is a friend or foe. And you can see it because I've blown it up now to show that T cell looking at that cell. And it'll make a fate decision. It'll either execute that cell and eliminate that cell, or it'll leave that cell alone and it'll go on and look for the next one. And that is the real biology of what T cells go on and do. So what we do at Anderson, and what that's done at Sloan, and what gets done at Penn, is essentially try and tip the balance in the favor of the graft versus leukemia effect, the anti-tumor effect. And that's where gene therapy comes in. So gene therapy is a concept that we're using, but in the laboratory. We're not putting genes, we're not doing therapy on a patient by themselves. We're taking the blood out of the patient and doing the genetic manipulation in the laboratory and returning those cells to the patient. And when we do that, we can tip the balance, because now you see the T cell that would be the native T cell that I would get from you today. And we introduce this CAR, this chimeric antigen receptor, and now the T cell has a retargeting molecule on it, just like Carl showed you. But it now can do something special where it couldn't do something before. And so if we return to the concept of transplantation, but in this case, we're doing a transplant from me into me. So this isn't from my donor, this gentleman in the front row, say, where he would be my donor. This would be from me into me, and that would be called an autologous transplantation, as opposed to an allogeneic transplantation. If you do an autologous transplantation, there's no chance of graft versus host disease. I don't have autoimmune disease. If I take my T cells out and I tickle them in the laboratory and return them to me, they're not going to cause graft versus host disease, because they have the same category, they have the same repertoire of T cell receptors that they had 10 minutes ago as when I drew the blood. But what we want to do is push the graft versus leukemia effect. And when we do that, we essentially have done it because we now have the chimeric antigen receptor on the right. You can see the little extra blue molecule that can recognize this B cell antigen, this leukemia antigen called CD19. It doesn't really matter. But for the purposes of this, we can essentially move my blood out of the body and return it with this essentially enhanced potential to target leukemia. And now you can see the little diagram and whether or not, and essentially we're ignoring the T cell receptor now because we put in a new immunoreceptor that essentially targets that B cell malignancy. And then that will make a decision to kill the tumor. So to bring all this into focus, we had to actually invent some different technology. Carl brought a technology with this virus work with HIV. And we actually thought of a different approach, where we didn't have to use viruses to genetically reprogram T cells. And actually, we use fish biology, believe it or not. And this is something that actually got invented by a gentleman at the University of Minnesota. But we recognized that this could actually have human application. So we use bits of fish DNA to reprogram human T cells. And then there are other tricks that we learned, and in fact, we learned them from Carl. And again, it shows you the power of cooperating and pulling together, where we learned how to grow these cells on these artificial antigen-presenting cells. Unlike the beads that Carl invented, we use cells as nurse cells that can tickle T cells to grow in an environment and grow them up. So here's an example now, and you can be a biologist with me, because you can see on the y-axis, massive numbers of T cells can be grown in log phase, and on the x-axis is time and culture. And T cells start out just with their own T cell receptor, and they end up having T cell receptor and this introduced receptor, right? So they can actually have this desired function to go after a tumor. And if this movie runs, you can see one of these engineered T cells. Go ahead and run the movie, where the green cells that are laid down on the plastic petri dish are the tumor cells that have CD19. Can you run it? Whoops, it'll get better in a second. And you can see that T cell, see it running around busy on the top there? It's trying to figure out if I'm going to kill that tumor cell or not. This is a reprogrammed T cell, and then you can see the tumor cell popped. And then you'll see another, that last tumor cell pop too. And the rest of course, the cells coming into the field of view have also essentially been interrogated by those T cells and killed by those T cells. So before we got going, we actually practiced in dogs. We practiced in pet dogs. My pet dog, your pet dog. About 1 million cases of lymphoma occur in companion canines in the United States. And for some families, we'll elect to give those canines CHOP chemotherapy. Basically the same chemotherapy as one would give a human being who had a particular type of lymphoma. And we figured out that we could actually help the dogs by not only giving them CHOP chemotherapy, but by adding back T cells in that context. And this looked to us quite promising because it was sort of this outbred model, and for all intents and purposes, dogs live with us, eat with us. My dogs, tend to sleep with us. And so they're very much part of the sort of the human continuum. And we were reassured by showing that we could actually shift the survival curve for some of these dogs by giving them T-cell therapy. So Carl alluded to the fact that this really is a partnership and is a team approach, and it really is. And in fact, this is my little organizational chart. And what we built at Anderson is to try and replicate what essentially is a little biotech company working in a not-for-profit superstructure. But all the components that you would recognize in the biotechnology industry, whether you'd have research and development and manufacturing, as well as your regulatory affairs team, your clinical trials team, all these groups of people have to come together. And this isn't funded by the NCI or the NIH, but they can't get their mind around this type of science. It's too, it doesn't fit the typical funding model. They want to see tight bench research, and this type of translational research falls into what's been called the valley of death, really, because it's very difficult for folks to, at least the federal government, to feel comfortable funding this type of science. But again, philanthropy, thank goodness, has really stepped in and recognized the power of doing this type of organization. So we've also been energetic and launched now 4 trials using this fish biology to reprogram the T cells. But we've taken a slightly different approach because because I'm a transplanter and my stakeholders are kids and adults undergoing transplantation. We've infused the T cells in that context. So we've married the T cells with autologous transplantation, with allogeneic transplantation, and increasingly now with cord blood transplantation. A lot of kids get cord blood transplantation, especially from ethnic minorities. And the data will be continue to come out, but I'll just show you one patient, just because it's interesting. And just to go to Carl's point, every patient tells a story. And again, it's very hard for us at the dawn of a new age to kind of figure out and make sweeping principles. But you can recognize that just by you don't have to be an expert in the field to recognize essentially that this is the amount of T cells that we can detect in the blood by this y-axis on the far left, and then days after the infusion. Something up to 6 months or so, this patient's out now. But for a long time, the T cells were tiny in number and actually disappeared from the bloodstream at one time point at 3 months, and then all of a sudden come back. Like, why was that? Was the patient threatening relapse? Was the patient with the T cells gathering in number and strength? Had the T cells found the right lawn to sit down in and now they could grow? All these questions remain to be answered. And we can plot other parts of the biology on the right-hand side where we can look at the T cells physically by seeing them under the microscope. Or we can measure essentially proxies for how the T cells are doing, whether or not there's any normal B cells in that patient, and they're not. Again, each tells a story. So we are also faced, like Carl's faced, with making sure this isn't just a trick. This can't just be boutique where we treat 25 patients a year. Basically, that's the number that we've enrolled. And we've infused now about 16 of those 25 patients. And that pretty much was a full-on effort at Anderson to get them to do that. A whole training program had to be put in place to train up the technicians, and it pretty much did me in to get those patients done. But 16 patients is great for those 16, right? But it's not going to move the field. So to move the field, we have to think for what the next part of it is. And we all have ideas, but one of the ideas we have is an idea that we can try and make T cells, not for every particular patient from their own blood, but choose one donor. So we'll choose Barbara as the universal donor. And we'll take her T cells and we'll put into all of you. And to do that, we've invented some technology that I think has some benefit at this point, where we've essentially want to make sure that there's no graft versus host disease. There's none of that unwanted destruction of the patient's normal body. But we want to preserve the anti-tumor effect with the chimeric antigen receptor. So we invented these little molecular scissors with a company called Sangamo Biosciences to take out genes. And we obviously have the fish in our trick that we can put into genes. So we can put genes in and take genes out. So again, this type of biology now is really right on the verge of being able to do these things. So I want to just leave you with a couple of just last thoughts. And that is, I love this curve. It's called the power law curve. And it's used a lot. It's how Google makes the money. It's how iTunes makes their money. And it's essentially, that describes a curve where you're looking at the number of drugs dispersed, but let's just flip it around. Let's look at the number of downloads, okay, on the y-axis, and then the number, essentially, of artists on the x-axis. So back in the day when we went to Virgin Records and bought Beyonce, right, there was just a wall of Beyonce on the shelf. And Walgreens and Virgin and whoever recognized that people like me were going to buy her, and she was a blockbuster. And that's how medicine has existed for decades in terms of these blockbuster drugs, where we really only have shelf space, quote unquote, for just a few drugs. But the problem is, whoops, I want to go. The problem is that increasingly cancer is becoming a biology of onesies and twosies. We recognize that individual patients have discrete problems that's related to their tumor, and we have to do better as physicians to design the drug that's going to target that tumor and leave the rest of the body alone. So we need to move our mentality away from this idea of these historical medical centers or historically how we've been doing things and start to get at where Google is and iTunes is, and that is the long tail. This, to go to the right, never touches, okay, zero. But it's all the onesies and twosies. It's all the little downloads you do of the obscure artists that essentially make it possible for Google to make money, for iTunes to make money. And why is that? Well, the reason is because what presses down on the power law curve and makes this curve asymptotic is the cost of distribution. When the cost of distribution of an electron went to zero, then iTunes made billions, Google made billions. And that's where we need to be as a field, because we need to get our costs of distribution down in terms of T-cell therapies, and we can. Because these T-cells have been made in academics, and we've lived on shoestring budgets for years. We understand, essentially, how to get things done with reduced costs. It costs about $12,000 right now to make our T-cell therapies, ignoring all the other back-end costs, which is just a fraction of what these other big quote-unquote blockbuster drugs cost. Carl alluded to it, $100,000 to get rituximab. So we can push these costs down, and we can get out now to where we essentially can look at these patients and individualize and personalize this type of immunotherapy so that we can be living out on the long tail. So I'm going to stop because we have to hear from Michelle. I had a lot more to tell you, but I don't want to lose you. But thanks for your attention.
Thank you, Larry. So I didn't expect this. That not only we got a lecture on wonderful science and medicine and clinical and so on, but also we got a lecture on economics. So the last speaker for the evening is Michelle Satherlane.
I hope that many of you like Andy Warhol, especially his Campbell soup cans or his Marilyn Monroe's, repetition. Or maybe for some of you it's the Goldberg variations. Because as you're going to see, you're going to hear 3 variations here on one theme. And the only person to blame for that really is Savio Woo, who's the chairman of the Scientific Advisory Board of ACGT, because 10 years ago, he and Margaret and others had the vision to support the kind of research that you're hearing about today. And the 3 of us here, Lawrence and Carl and myself, actually were the beneficiaries of the largesse of ACGT and did what we're showing you today with these monies. So this is going to be variation 17 or 18. You know that they get harder and harder before the last one. And so after Carl's masterful, really, introduction of what CAR therapies can do and how to develop them in the future, followed by Lawrence's more granular description of bone marrow transplantation and the underlying biology, I'm going to take you to the next variation and really focus more on the engineering and the CAR, and to try to explain to you what's been accomplished and what some of the challenges are going forward. So the next slide, that's for me to, yes. So we're all interested in developing effective cancer therapies. And these would have to be powerful enough to not just slow down the progression of cancer, but actually kill off and eliminate the cancer cells. But such potency has to be constrained, and it has to be safe, because if your drug or your therapy has too many side effects, this will, of course, limit your ability to treat the underlying disease. the key to resolving many aspects of potency and safety is specificity. If your therapy is very much targeted to the cancer, distinguishing the cancer from the normal cells, so if it's highly specific, then perhaps you can reconcile potency and safety. It's not easy to do, and there are many drugs that are not very potent but safe, and others that are very potent but not so safe. I think all of us at this table have been mesmerized for many years by the immune system, which can do just that. That is to say, specifically recognize a virus or a bacterium and eliminate it with hardly any collateral damage, if you like, to normal tissues. So harnessing the immune system is really what really armies of researchers have tried to do for a long time. Now, that's not been so easy. And if you look at a review in a fine journal just 10 years ago explaining how one can harness the immune system to fight cancer, you'd find these two major approaches. One is the so-called vaccine therapy, by analogy to infectious disease, trying to stimulate in the body a vigorous immune response that will attack the cancer. As it turns out, that's not so simple, because viruses are very different from us. And so the immune system can tell them from our own cells, but cancer is really our own cells. They're really a part of us, and so that requires a much more subtle distinction between the target and the normal tissues, which the immune system has trouble achieving. So cancer vaccines may still work one day, someday, but it's fair to say that till now they've met with rather limited results. Another approach, which was pioneered at the National Cancer Institute, starting in melanoma, a tumor that's accessible to the surgeon because it's under the skin, and that, it turns out, is rather immunogenic, led to the development of what's called tumor-infiltrating lymphocyte therapy. So in this approach, starting from a surgically excised tumor, the tumor could be brought to the laboratory, and the T cells that were there, which logically you would think include some T cells that are capable of recognizing the cancer, would be expanded in bags and then infused back to the patient. And there's a fair fraction of patients that actually respond to this therapy. So that taught us a lot. But it also pointed out two major limitations. First of all, even in melanoma, you cannot grow out these T cells for every patient, only some of them. And furthermore, and more importantly, you can't do this for many, many other cancers, for which you can't access a surgical specimen or for which there aren't that many T cells to grow. So clearly, this taught us that T cells could do a lot, and so did at about the same time the bone marrow transplants that Lawrence told you about. But we needed a new way to make T cells that would attack cancer. And so this is the grand schema, the biological underpinning, if you like, for engineering immune defenses. The major hypothesis is that T cells can be genetically enhanced, that is to say, to acquire supraphysiological, non-natural properties to overcome the defense mechanisms that tumors utilize to fend off immune responses. And in doing so, there are two main objectives. One is to make the T cell recognize the cancer. So that's an issue of specificity, and for that you need to identify an appropriate target, a molecule that's on the surface of the cancer. The second thing that you can do when engineering T cells is to increase their functions so that they will acquire properties that are more than the usual, if you like, and will enable to reject tumors. And this brings us to the CARs that Carl and then Lawrence already introduced to you. And I'll just give you the schema of these and emphasize the three key parts in these CARs. There are three key constituents in the structure and three key functions. First is on the imagine a cell that's here. This is protruding outside of the cell. This is the part of the receptor that will latch onto the tumor. So one key function is to recognize a molecule that immunologists just call an antigen. Upon the binding of this T cell to the tumor through this end of the molecule, an activation signal is transduced or is given to that T cell. It tells the T cell to get angry, to do something about it. You've just found a target, do something about it. And there's a third part in this CAR which is also incorporated into this intracellular portion, which gives a more subtle instruction to the T cell to proliferate so that it will divide more, so you will increase the size of this army, and also to exert different functions. So really, three key parts in this CAR. A target that we were interested long ago and studied before submitting our first grant to ACGT is one that you've already heard from today. It's called CD19. CD19 is the molecule that's on the surface of the majority of leukemias and lymphomas. And as important as where this molecule is found is where it is not found. And remember the problem of specificity, of singling out the tumor without doing harm to normal tissues. Well, this molecule is expressed on some normal cells. They're all the B cells that we have in the body. But very importantly, it's nowhere else in the body. So if you were able to make a T cell that eliminated, that eradicated any cell in the body that possesses this CD19 molecule, you would predictably destroy all the normal B cells, which is not ideal, but can be handled in the hospital, but nothing else. And that's much better than what conventional chemotherapies can do. And so it's already been 10 years that we showed in mice. This is actually 10-year anniversary of this paper. This is a mouse in which we've introduced human leukemias. They go in the bone marrow. You can see here along the spine or in the calvarium of this mouse. It's a lateral view of a mouse, imaged with FDG like we use in some cancer patients. And here we give them a single intravenous infusion of T cells, human T cells, that have been genetically engineered to recognize CD19. And this shows you that half of these mice are long-term survivor. If you give more T cells, you can actually cure all of them. And so these data certainly were the stimulation for that ACGT grant in 2004, and also doing the clinical trials. Now, to do that, as Carl already mentioned, you have to establish a manufacturing process. Lawrence also alluded to the challenges that this poses, and these were resolved at Sloan Kettering by my colleague Isabelle Rivière, who developed this process, starting from the patient's cells that are collected in our collection room. The T cells are activated using actually the beads that Carl had originally described. These cells, then, we mix the vector that carries the gene into these cultures. They're in these bags here, about 1 liter bag, about this size. And this culture lasts 10 days or so. In that time, the gene is introduced into the T cell, and the T cell starts making this molecule that I showed you on the surface of the cells. And after 10 days, the cells are ready for infusion. The trial that I want to tell you about is one that we started in adults with acute lymphoblastic leukemia. And it was led by my colleague, Rainier Brentjens at Sloan Kettering, now taken over by Marco Davila. And this is the table from the first report that we published earlier this year. And that, too, was another one of these New York Times cover stories. And so here are the 5 patients that we treated first. Individuals from age 66 to 23. They all had ALL. They all received an initial chemotherapy, after which they go, most of the time, into a complete remission. Call that CR1. Some of these remissions last as little as 5 weeks. Others may last, as you see here in this example, 3 years. And then, unfortunately, unlike a situation in children, most of these patients will relapse. Some of them get a bone marrow transplantation, which can actually cure a small fraction of these patients, but many do not get a transplant and eventually relapse, at what point they receive what's called salvage chemotherapy. As the name suggests, this is an aggressive therapy that you do as a last hope that you can eradicate tumor a second time. And this rarely succeeds in adults. And in fact, these are the patients that we enrolled on this trial. And as you see, all but one, which we found out about later, all of them had a lot of leukemia a few weeks after that salvage chemotherapy. The punchline from this study is shown in the next column, which is that all of them, 4 out of the 4, became negative for the disease, and I'll show you how we know that and how quickly this happened. This is marrow that I know you can't tell. This is a picture of the bone marrow where the leukemia lives in these patients. These are the 2 most spectacular patients. 60–70% of their bone marrow, all those little purple dots, are infiltrating leukemia. The marrow doesn't work anymore. You know that from our marrow, we make red cells, white cells, platelets. These individuals need transfusions. They're at risk of infections. They need platelet transfusions so that they don't bleed. That bone marrow is chock full of leukemia. Within a few days, it's gone after a combination of 1 round of chemotherapy in the T cells. And as you see, you wouldn't know this, but I'll take my word for it, please. After 2 months, this marrow looks normal again and free of any tumor. It's actually not quite normal. When you look closely, and these graphics, I won't even go through them, but this shows you that everything is normal except that they have no B cells, because the B cells express CD19. So the leukemia appears to be gone, and so are the normal B cells. Now, we can take a look in greater detail using modern molecular techniques called deep sequencing. And if we do that, this is the most sophisticated method that's available to detect very small amounts of leukemia that may still be in that marrow. We found that it decreased to undetectable levels in all these patients. Day -1 of treatment, these patients had 58 of these hits on this sequencing, 0 by day 55. This patient, I'll skip, had none to start with. This individual, you see 2,001,000 at day 11, 3 weeks 600, 2 months 0. This patient, ALL05, the most spectacular perhaps, 3 million of these sequencing hits at 1 day before the T-cell therapy, 0 as early as 8 days later, and that's maintained for several weeks here. Same thing in patient 5. So what we see is that there is not just what's called a complete remission, but what hematologists call a molecular remission. That's the best response that you can hope to get in patients like this. So in our studies, we then give the standard of care to these patients, which normally would be a bone marrow transplantation. At the stage of their disease, these patients were not even eligible for that. You wouldn't even propose that because it's guaranteed to fail. However, it is known to treat patients, so we follow this treatment by a bone marrow transplantation, and that's something we may discuss, maybe not, later. So I just want to update you from this paper that was in March, and by now we've treated, actually 3 weeks ago we were at 14 patients with adult ALL. We're now at 16, but just looking at where we were at 2-3 weeks ago, 14 patients were treated. 3 of them had this scary Philadelphia chromosome, and those two responded very well. And in fact, 12 out of these 14 now have achieved a complete remission and even a molecular remission. So the results are holding up as the number of patients increases. So in Memorial, what are we doing now? Well, we're expanding the trials targeting CD19. We have trials in chronic lymphocytic leukemia, including a collaborative study with Carl at the University of Pennsylvania, comparing his CAR and ours. We have a study in patients who have relapsed ALL, which I just described to you. 29 patients enrolled, and 14, or in fact 16, treated by now. We're now moving our CLL therapy to an earlier stage of disease. We're also giving this therapy in pediatric patients using a different kind of T cell from a donor. This is not the patient's own T cells. We're also starting a study now in lymphoma, another disease that expresses CD19, and recently treated 2 patients who also have complete remissions, actually, and are now starting a different study in ALL in children, quite similar to what Carl is doing at the University of Pennsylvania. So this is the model that all of us are now pursuing in this new approach to medicine. This slide shows you how this proceeds, which by now I'm sure you're familiar with, but also highlights the differences between the studies. Sure, we all enroll patients on these trials, but even for the same disease, they don't always have quite the same severity of disease or the same status. In everybody, we collect those cells and activate those cells to introduce the genes, although we use different methods to do that. We then use different chimeric receptors. They are introduced using different methods for gene transfer, and in case you didn't quite catch that, Dr. June uses a lentiviral vector, Dr. Cooper a fish-derived vector, and we use a mouse-derived vector. So plenty of variation there. The cells are then expanded using different methodologies, and we give different doses at different centers. Some people give a dose escalation like we do. Others give whatever you get in a 10-day culture, and other people give the same dose to everybody. And so the point that I'm making here is that we're just at the beginning of this. There are many variables, and it's only through the support of multiple clinical trials at multiple centers that collectively we're going to get to a rational understanding of how this exactly works and how to optimize it. So this is just reiterating what I just said. Different CARs, different vectors, different manufacturing, different final product, different chemotherapies, and of course different patients. So for all of us, it makes it quite complex to really analyze these data, but it's coming along very nicely. And in fact, the world of CARs is expanding, and you remember this first diagram that I showed you? This is just another sample of this. You can just tell that there are many molecules now that investigators at different centers are comparing, and again, we need to know which ones are really better than others. And that will take more studies at more centers, but it's all very feasible. I just want to allude to some of the challenges that face the field. Both Carl and Lawrence emphasized the need to develop the infrastructure for manufacturing these cells and providing them to more patients. I agree with that. I'll give you one little twist on that, another variation here. But I want to address two other aspects. In some diseases, we know that one CAR can work very well. The one we have works very well in ALL, acute leukemia. It's less effective in chronic leukemia. It's the same molecule, it's the same target, and yet in two different diseases, the outcomes are different. So from this and many other examples, we know that we have to modulate the strength of these T cells. And Carl alluded to that, different potencies that we can now engineer in these T cells. We may want to make cells that persist for weeks. Maybe we want them to persist for months. We don't quite control that fully, but we're very close. One issue that I want to raise your attention to, for which we clearly need more research and more really philanthropic support, is the fact that we want to extend this to many other cancers: prostate cancer, breast cancer, colon cancer. But for that, we need the right target. So CD19, which I emphasized a little bit, has almost perfect attributes. But there are very few perfect targets for most of the other cancers. So how are we going to do that? I'll say one thing about that, and then one thing about how we're exploring a novel approach to make these T cells for infusions to patients. So anyway, how can we modulate the potency of these cells? That's a very complicated topic, but I'll use a cartoon that I use for graduate or medical students to show you what we can do. This chimeric receptor is on a T cell, and here's a tumor cell, and it's binding, you remember that, latching on to this antigen. It could be CD19 in the example I showed you before. This then it activates the T cells, which gets angry, and additional signals take place soon after the initiation of this response. Well, there's a number of changes that occur on the cell surface. Some receptors that were not present on the surface now appear, others go away. There's a very complicated tangle, really, that takes place. So we're learning now how to design some of these receptors that will specifically act on these molecules that appear and disappear on the surface of the T cell as the immune response proceeds. And therein for sure lies the key to making more powerful T cells. Another problem that I said I would mention very briefly is that of finding the right target. Well, unfortunately, for a lot of cancers, we don't have a molecule that is just on the cancer cell and not present on any other normal cell. Unfortunately, most of these candidate targets are found on both tumor cells and some normal cells. And these cells are so potent that they may not distinguish the two. So they may eliminate the cancer, but they'll also cause damage to the normal tissue. One study that showed that, and there are several recently, was a study in colon cancer where a group selected a particular target, and there seemed to be a response of the tumor, but that target was on normal cells in the colon, and there was irreparable damage to the colon. It was reversible eventually, but they had to stop the therapy. So we need targets. So a lot of scientists now, I think, will be looking for molecules that could be unique to the cancer. But what if there aren't unique targets on at least some of the cancers? Well, are we then stuck dead in the water? Not at all. And there are strategies, and this one's quite complicated, but I'll just try to summarize it briefly. If you can make a smart T cell that only attacks tissues that express two molecules, not one. Normally cells see one target, and when they see it and bind to it, they do their thing. But we can engineer T cells now that will only act if they see two molecules at the same time. And so if we can intelligently pick those two, even though neither one is unique for tumors, because A is also found on some normal tissues, and B, which is on this tumor, is also found on some other normal tissues. Well, if we make a smart T cell that only attacks tissues that express A and B, we may now create cells that will spare the tissues that express B alone or A alone. Sorry for the science on this variation, but this perhaps will give you an idea of how scientists try to create selectivity for the tumor, even if there isn't actually a unique target. The last example or a future direction I want to show you is, it's a bit of a crazy project. It's very futuristic, and I don't know actually if this one will ever see the light in the clinic, but it may. Carl alluded to the huge challenge of making large amounts of these T cells. And I think it was Ford, the example, the T Ford. So the big assembly line and make lots of personalized cells. And that's certainly the approach that's the most closest at hand. Lawrence showed another approach where you can take from some donors T cells and remove a receptor that may cause harm in the recipient and put the one you want in. So as he said, you cut one out and put one in and give those T cells. There's another one that I don't think this slide is very clear, but I'll just say that if we can immortalize these T cells in culture, and we do that by converting them to what's called a pluripotent stem cell, something that a Japanese scientist described a few years ago, and he got the Nobel Prize actually, for describing this way of making cells that could acquire the properties of the most primitive stem cells. The advantage of that is that these cells you can grow and grow and grow and grow indefinitely. And so we asked, can we make from those T cells in a dish T lymphocytes? And of course, in T cells, in those cells, we introduced these chimeric antigen receptors. And that's what we recently published, actually just this month. It's a method to make a T cell in a dish. So we're not ready to do this in clinical trials. There's a lot of science that needs to be worked out. But we're hopeful that for sure we'll learn a lot of biology, and perhaps we may be able to not just make the CARs, but actually in synthetic fashion make the whole T cell, something that's indeed very futuristic, but may work. Both Carl and Lawrence referred to the dawn of a new age. This is my last slide. And well, 10 years ago, 15 years ago, there were some reports showing that you could put what's now called a CAR in a T cell and redirect and make it kill something in vitro in culture. But that didn't work in mice and doesn't really work in humans either. What we've seen over the last 10 years is augmenting the potency of these T cells. These T cells survive, they proliferate in the recipient. An army of those can be grown in culture and then persist in the patient's patient. We have learned a lot about how to make them overcome some of the barriers in the microenvironment, the environment of the tumor. And I think we're learning now which are the better T cells to reprogram, which means there are fewer to make. If they're better, you don't need to manufacture so many. And that's the sort of the Iron Age. And the Golden Age that I would see is one where we would find ways to make very potent T cells that are very specific, that do not attack normal tissues, that do not engage in some of the dramatic responses. And Carl alluded to one example of that little girl who had these tremendous fevers for several days. Of course, that was very beneficial. It was actually the signature that the T cells are working. But adults can't, and even children cannot handle responses of such a magnitude. So we have to learn to restrain this power and make sure these T cells persist but don't become monsters either. We don't want cells that are growing out of control. All this has to be done in a way where we really control the fate and number of these T cells. The last slide is just to acknowledge too many people, but one I mentioned already, my colleague Dr. Rainier Brentians, who's the principal investigator on many of our trials. Isabelle Riviere, that makes the T cells for all the studies at Sloan Kettering. And of course, the Alliance for Cancer Gene Therapy, that really was the first organization to actually fund our program. Thank you.
Thank you, Michel. Before we go on to the question and answering period, as it's been clear from all the speakers, that none of this would have happened, fantastic results, to see all these patients responding and so on, would have happened without the work of two people, and that's Edward and Barbara Netter. Ed was no longer with us. Barbara, will you please stand? And of course, for all these years, even though everybody said, oh, Savio did this, Savio did that, well, Savio didn't do nothing. Everything was done by Margaret Cianci, who was the executive director of ACGT. As you have heard from the three wonderful speakers, this new field of immune therapy for cancer using gene and cell therapy, even though there are all sorts of variations, this and that, different genes, different vectors, different cells, or whatever, okay? The bottom line is that they all have they all share a common theme, and that is they empower the immune system in our body to recognize the cancer and kill them. Basically, they're giving the immune cells, the killer immune cells, a zip code, so that they can go back there and just target the cancer and kill it. And because of that, we have tumor selectivity, which is different from just good old chemotherapy, okay, which kills a lot of other cells. And because of that, we have relative safety, because we're killing mainly cancer cells and not the normal cells. And because of that, we have effectiveness. You can see the patients are responding. Tumors went away, 6 pounds of tumor, my goodness. I mean, and it just dissolved, right? It's never been seen before. And finally, even more important to me is that the effect is lasting. We are talking about having these killer T cells, killer immune cells circulating in a patient's body that will recognize cancer specifically, and they continue to persist in the patient's blood, so that whenever there is new cancer coming of the same type, they will be killed, so that we don't see the relapse as often as we normally see with chemotherapy and other types of traditional therapies. So this is a new field that is going to be so promising and exciting for all sorts of tumors, And as you have heard from Dr. Satterlane, one of these days we're going to go from dumb phones to smartphones, and now we're going from dumb T cells to smart T cells. So we all look forward to that day.